Welcome to Aquatic Biology. Today, we are diving into the fascinating world of lake morphometry. Imagine trying to fill a shallow kiddie pool versus a deep, narrow water well. Even with the exact same amount of water, they behave completely differently under the sun and wind. In nature, lakes act the exact same way. By the end of this video, you will understand how the physical shape and dimensions of a water body dictate its entire biological and chemical behavior. Let us get started. Now that we've introduced the topic, let us define what this term actually means. The word morphometry comes from two roots: morpho, meaning shape, and metry, meaning measurement. Therefore, lake morphometry is the scientific measurement and mathematical analysis of a lake's physical dimensions. Scientists map the three-dimensional shape of the lake basin, including its surface area, total volume, and depth profile. They gather this data by creating bathymetric maps, which are essentially underwater topographic maps showing the contours of the lake bottom. This gives us a complete physical blueprint of the water body. Building on that definition, you might wonder why a biologist cares about physical measurements instead of just studying the fish. The truth is that the shape of a lake completely controls its biology, chemistry, and physics. For instance, a lake's depth determines whether it will form distinct temperature layers, a process known as thermal stratification. This directly affects where fish can survive. Furthermore, knowing the exact volume of a lake is absolutely critical if a toxic spill occurs, because it allows scientists to calculate the exact concentration of the pollutant. Without these measurements, we are blind to how the ecosystem functions. Having seen why morphometry is crucial, let us look at one of its most important measurements: the mean depth. While the maximum depth only tells us about the deepest single point, the mean depth gives us a much better picture of the lake's overall character. A lake with a very large mean depth will generally be colder, less productive, and have clearer water. Conversely, a lake with a small mean depth will be warmer, grow more algae, and support a massive amount of plant life. It is a fundamental indicator of how the lake will behave ecologically. To quantify this concept, we use a very straightforward mathematical relationship. The formula for mean depth comes from taking all the water in the lake and spreading it perfectly evenly across its entire surface area. In this equation, z bar represents the mean depth, measured in meters. The variable v stands for the total volume of the lake in cubic meters, and a represents the total surface area in square meters. Physically, if the volume increases while the area stays the same, the mean depth increases. But if the area increases for a given volume, the water spreads out more, and the mean depth decreases. Before we move on, let us see this with a concrete example. Imagine a local reservoir providing drinking water. We are given a total surface area of 2 million square meters and a total volume of 15 million cubic meters. We need to find the mean depth to see if the reservoir will heat up quickly in summer. We choose our mean depth formula because it directly relates volume and area. By substituting our known values, we divide 15 million by 2 million. The units cancel perfectly, leaving us with a final answer of 7.5 meters. Physically, this relatively shallow depth means the lake will likely heat up quickly, potentially leading to algae blooms. Now that we understand depth, let us shift our focus to the edges of the lake with the Shoreline Development Index. This index tells us how irregular or winding the shoreline of a lake is compared to a perfectly round circle. A perfectly circular lake has the smallest possible shoreline for its area, meaning fewer shallow edges for plants and fish. However, a lake with a highly irregular branching shoreline has a massive amount of edge habitat, known as the littoral zone. This index helps biologists predict how much biological activity will happen near the edges of the lake. To measure this edge habitat, we use the Shoreline Development Index formula. Conceptually, this formula is simply a ratio comparing the lake's actual shoreline length to the shortest possible shoreline it could theoretically have if it were a perfect circle. D sub L represents the index, which is a dimensionless number. L is the total length of the shoreline in kilometers, and A is the surface area in square kilometers. The denominator represents the circumference of that ideal perfect circle. As the shoreline becomes more jagged and irregular, the length L increases, causing the index to become much larger than 1. Let us apply this formula to compare the habitat potential of a natural lake. We are given a lake with a surface area of 3.14 square kilometers and an actual shoreline length of 12.56 kilometers. We want to find the index to determine the available edge habitat. We plug our values into the formula. Inside the square root, we multiply the area by pi, which gives us approximately 9.869. Taking the square root and multiplying by 2 gives a denominator of 6.28. Dividing our actual shoreline by this ideal circumference gives a final answer of 2.0. This means the shoreline is twice as long as a perfect circle, providing double the shallow water habitat for fish to spawn. Now that we have used this formula, let us understand where its denominator comes from. The denominator claims to be the circumference of a perfect circle with the exact same area as our lake. Let us prove this mathematically. We start with the standard geometric formula for the area of a circle, which is A equals pi times r squared. Our goal is to find the radius, so we can eventually find the circumference. We divide both sides by pi to isolate r squared. Then we take the square root of both sides. This gives us an expression for the radius of our hypothetical perfect circle purely in terms of its area. Continuing from our expression for the radius, we can now find the ideal shoreline length. We take the standard formula for the circumference of a circle, which is c equals 2 times pi times r. We substitute the radius expression we just found into this circumference formula. Now, we bring the pi from the outside into the square root. When pi goes inside a square root, it becomes pi squared. The pi squared in the numerator cancels with the pi in the denominator, leaving us with exactly 2 times the square root of pi times a. We have successfully proven that the denominator in our index formula is indeed the exact circumference of a perfectly circular lake. So we have proven how the math works. This brings us to a visual summary of these concepts. On the screen, you are looking at 2 distinct diagrams. At the top, we have a top-down view comparing 2 lakes with the exact same surface area. The blue circle on the left has an index of exactly 1, showing the minimum possible shoreline. The jagged, snowflake-like shape on the right has an index much greater than 1, highlighting the massive increase in edge habitat. Below that, a 3-dimensional cross-section of a lake basin shows the flat surface area at the top, the deepest point labeled as maximum depth, and a dashed line indicating the much shallower mean depth. This perfectly illustrates how the physical bowl dictates the water's distribution. To wrap up our lecture, Let us review the most important ideas we have covered. First, lake morphometry is the essential foundation for understanding any aquatic ecosystem. You cannot understand the biology without knowing the physical bowl that holds it. Second, the mean depth is a powerful predictor of a lake's temperature profile and overall biological productivity. Finally, the Shoreline Development Index provides an objective mathematical way to measure how much vital edge habitat is available for aquatic life. You have now mastered the core physical measurements that allow biologists to protect and manage our precious freshwater resources effectively. Great job!